Hello, everyone. Thank you for joining us today for one more CMDI summer seminar. Today we have Catherine Armbruster. She got her PhD at the University of Washington, co-mentored by Matt Parsek and Luke Hoffman. She currently is working as a postdoc at the Bomberger Lab in Pittsburgh, studying the evolution of Pseudomonas aeruginosa in cystic fibrosis lung infections. As a reminder, please, if you have a question, just type it in the chat or raise your hand in the little chat too, and then we will we will address the questions at the end. Catherine, thank you for joining us, and we're looking forward to hearing your talk.
Okay. All right. Thanks, Juan. I'm going to start sharing my screen, so let's get this to work. All right. All right, one second. Oh man, my sorry, my button that I normally push is not visible right now. Okay. All right, can you guys see that? I'm assuming.
Perfect.
Yep,
Okay.
that works.
Yep.
All right, thanks, thanks, guys. All right, so of course I want to thank the organizers for inviting me. I was telling them earlier in this call that this is like the most exciting thing I've done in a couple weeks. I've just been sitting at home, social distancing. So today I'm very excited to talk to you about the work I've been doing as a postdoc with Jen Baumberger at the University of Pittsburgh, where I'm studying the evolution of Pseudomonas in the upper respiratory tract of people with CF. And so my title is really aspirational today. It's the past, present, and future of Pseudomonas evolution in the sinuses of people with CF. And I'll tell you a little bit more about what I mean by the past, present, and future in a couple slides. So stay tuned for that. So in GenSob, we study cystic fibrosis, which is a genetic disorder that's caused by mutations in the CFTR chloride channel. And so historically, people with CF died in infancy due to GI issues, but this aspect of the disease is better able to be managed now. And so now that people with CF are living longer, the majority of morbidity and mortality is caused by lung disease. And so here's just a little cartoon of to summarize what happens in CF. So when there are mutations that inactivate CFTR, it results in a buildup of thick, sticky, acidic mucus in the airways. And this unfortunately ends up being a great place for bacteria to grow. And it's thought that it's a combination of these bacterial infections plus the robust but ineffective host immune response that causes the devastating damage to host tissues that ultimately either requires a lung transplant or results in the death of the patient in CF. And so in particular, Pseudomonas aeruginosa is a dominant pathogen in CF, and it's associated with a huge amount of morbidity and mortality. And so that's what we primarily study in GenSob, and that's what I'll talk to you guys about today. And so we already know a lot about the kind of the course of a Pseudomonas lung infection in CF from decades of research actually on this topic. And we know that Pseudomonas evolves over time during these decades-long CF lung infections. And so this figure I'm showing you here is going to summarize how we know that there's kind of different stages of Pseudomonas lung infections in CF. So early in life, people with CF are not colonized by Pseudomonas typically, and so maybe there'll be an initial sputum sample that is culture positive for Pseudomonas early in life, but antibiotics can usually knock it back, and that's maybe the initial stage of infection there. Then there's this intermittent stage of infection where throughout childhood, maybe occasionally there'll be sputum samples that are culture positive, but again, the antibiotics can be used to treat this infection. And then usually around late teenage years or early adulthood, people with CF and Pseudomonas will typically transition to what we call the chronic phase of Pseudomonas lung infection. And at this point, the amount of Pseudomonas in the sputum is not responsive to any therapeutically achievable concentrations of antibiotics. And during this period of time, this chronic Pseudomonas lung infection, there's a series of really predictable events that happen, and I've circled them here. And this is what we call pathoadaptation, and that's what I'm going to talk to you guys about today, and I'll define it a little better in a couple slides. But it's this adaptive evolution that occurs leading up to and during this chronic phase of infection. That's what I'm going to talk to you guys about today. So it's a couple of the things that are summarized in the slide here are the clonal selection of a single clonal population of Pseudomonas in the lungs during chronic infection, mutations that promote biofilm formation. Biofilms are dense aggregates of bacteria encased in a protective extracellular matrix. And then there's a significant amount of genotypic and phenotypic adaptation that happens during this time as well. So I want to just give you guys a little more background on what I mean by pathoadaptation, because I know that's a term that is maybe unclear or that folks haven't heard necessarily before. So what are the pathoadaptive traits that we know Pseudomonas develops, at least based on the decades of literature that we have now about lung isolates of Pseudomonas, so Pseudomonas from the lower respiratory tract of people with CF? Well, we know that during this chronic phase of Pseudomonas infection, there's this long-term persistence of a small number of clonally related lineages in the lung. So within a single patient, they'll typically have one or very few genetically unrelated Pseudomonas isolates. And so this is just a figure on the right from an early paper that's showing longitudinal Pseudomonas isolates from a single patient, and they're doing PFGE, which is a molecular fingerprinting technique, and they're showing that these are all clonally related isolates in this patient. And so despite these typically being clonal populations of Pseudomonas during chronic CF lung infection, there's high levels of phenotypic diversity. So this figure that I'm showing here is from a paper by Peter Jorth when he was postdoc with Pradeep Singh, and is showing a kind of a suite of these classic CF pathoadaptive phenotypes. There's in vitro tests that you see in most CF Pseudomonas papers to test for these pathoadaptive traits. And so why do we care about these pathoadaptive traits and these phenotypes, aside from the fact that they're really easy to do in the lab? Well, there's actually a huge amount of interest in these phenotypes because we think that they could potentially be good, useful biomarkers for example, what stage of disease a person is in, or potentially even maybe if there's somebody might be likely to exacerbate in the future or not. And so here's just one paper. This is from a Seattle group, Luke Hoffman's group, and it's showing down the column is a whole bunch of different classic Pseudomonas pathoadaptive traits, and then they're showing the predictive power of those traits in distinguishing between the new onset of Pseudomonas infection, intermittent infection, and chronic infection. You can see towards the top, there's a couple of phenotypes there that do a pretty good job of predicting which stage of disease somebody would be in. So clonal population of Pseudomonas, high levels of phenotypic diversity, and strong mutational parallelism. What that means is that even though people with CF will have a different genetically unrelated Pseudomonas strain from each other that's colonizing their lungs, the course of this infection plays out really similarly, and in terms of the evolution and the mutations that this lineage of Pseudomonas will acquire over the decade or so that it's in this person's lungs. And so this is just one example, this figure from Eric Smith, where you can see that there's a whole bunch of genes that got mutated in these populations of Pseudomonas from the CF sputum that they were following. And they saw, for example, this MEX-Z transcriptional regulator of efflux pumps was mutated in 18 patients. And so that's what mutational parallelism means in this context, is that the same genes are getting hit in multiple patients. So suggesting there could be some common selective pressures there. And then there's mutations in global regulatory genes. So Pseudomonas is exquisitely capable of sensing and responding to its environment, and has a bunch of signal transduction systems that, for example, mediate the transition from kind of an acute mode of growth to expressing chronic infection phenotypes. And here's just one paper where they found this red S sensor kinase that normally what it would do is allow Pseudomonas to transition between acute infection phenotypes like expressing the flagellum and toxins to chronic infection phenotype like overproducing biofilm. But in their paper, they figured out that mutations in this sensor kinase locked Pseudomonas towards the biofilm mode of growth. Another common kind of pathoadaptive phenotype or trend in the CF lungs is the emergence of hypermutator Pseudomonas strains. So these are Pseudomonas isolates that have mutations in DNA repair genes, and they get way more mutations than non-hypermutator isolates. Okay, and so that was, I wanted to just kind of get everyone on board with what I'm talking about with pathoadaptation based on this huge, rich literature that already exists on using longitudinal collections of Pseudomonas isolates from the lungs. And now in Jen's lab, when we're thinking about how do we block potentially these chronic lung infections from developing with Pseudomonas, we thought of two opportunities where we might be able to intervene. And one of them is maybe if we could block the initial acquisition of Pseudomonas in the lungs, that would be a good opportunity. And then another possibility is maybe if we could block that adaptive evolution that seems to be important for persistence in the lungs, and maybe that's another opportunity. And in thinking about both of these, we actually shifted our focus to a different site in the respiratory tract where people with CF have chronic Pseudomonas infections, and that's the sinuses. And so chronic rhinosinusitis, or CRS, is incredibly common in CF, but not very well studied, especially not compared to the lower respiratory tract infections. But people with CF get CRS for the same reasons, largely that they get lung infections. And so our inspiration to focus on the sinuses actually came from literature that was originally intended to link allergic rhinitis in the upper respiratory tract to asthma in the lower respiratory tract. And so it's this unified airway hypothesis that proposes that we should be thinking about the respiratory tract as a single unit from upper to lower respiratory tract. And so in thinking about this, we wondered whether we could target Pseudomonas populations in the sinuses in order to prevent or delay chronic lung infection. And there's actually a lot of literature that that supports this idea. So actually, the possibility that the sinuses can be a reservoir for transmitting bacteria into the lower airways in CF. And so a prevailing hypothesis in our field as to how Pseudomonas even gets into the respiratory tract to begin with is that Pseudomonas could be inhaled from the environment and first take hold in the sinuses before reaching the lungs. We know that people with CF can aspirate nasal secretions into their lungs. There's also evidence from a bunch of different pediatric studies of culture positivity of Pseudomonas in the upper respiratory tract before the lower respiratory tract. There's microbiome data that supports this possibility that the sinuses, changes in microbial communities in the sinuses, can drive changes in the lower respiratory tract as well. And there's also been some genotyping, kind of early genotyping studies that have seen the same genotype of Pseudomonas present in the sinuses and lungs, but there hasn't really been a good genomic study yet that we're aware of. Okay, and so with this in mind, we came up with this hypothesis that Pseudomonas could potentially be pathoadapting in the sinuses even before reaching the lungs. And so we're wondering whether environmental Pseudomonas inhaled into the sinuses might first evolve, and the sinuses could act as kind of a training ground for Pseudomonas to get used to the respiratory tract before it eventually reaches the lower, the lungs in the lower respiratory tract. And so the study that I'll tell you about today is actually just on the sinuses, but we have a series of ongoing studies in the lab now in adult and pediatric cohorts that all kind of test the same overarching hypothesis and model. And so this study was a big collaboration between basic scientists and clinicians, and I just especially wanted to shout out to Vaughn Cooper, who's a evolutionary biologist that's here down the hall at Pitt. And he was hugely helpful, as well as Chris Marshall, who's now at Marquette University with his own lab. And then our main clinical collaborator is Stella Lee, and she's the director of the CF Sinus Clinic here at Pitt. And so this is our first foray into a clinical study in Jen's lab, was the study of the sinuses. And Vaughn actually liked to say to me early on when we first started this study that by studying adults here in CF, it's kind of like we're coming into a movie halfway through the movie. And so this is where the past, present, and future from my title comes in. And so imagine if our 2-year study of the sinuses of adults with CF is kind of like watching, 30 seconds of the middle of a movie, and we don't know what happened early on in the movie, but we can guess using comparative genomics and population genetic approaches. And then we also don't know what's going to happen at the end of the movie, but we can also make some educated guesses there. And so this is what I meant by the past, present, and future of the sinus infections for the talk today. All right, and so here's our chronic rhinosinusitis study. It was a 2-year longitudinal study of 33 adults with CF and with chronic rhinosinusitis. And so these people came in every 3 months to our sinus clinic here at Pitt. And then they also came in if they were experiencing a sinus exacerbation. And so at every time point, we collected a bunch of patient clinical data that's actually been recently published by Anna Zemke, who is another researcher here at Pitt. And I just wanted to kind of summarize a couple of the key points from her paper for the early analysis of this cohort. And especially I want to draw your attention to the age of the patients that I'll be talking to you about today. So on average, the median's about 31.5 years old. And so really we are kind of coming in halfway through the story. These folks could have had Pseudomonas for 10 years at this point. And a key finding from Anna's paper was that sinus exacerbation increased the odds of the next visit, the next time that somebody came into the clinic, the CF clinic here at Pitt, that would be a pulmonary exacerbation. And so this was actually really encouraging and interesting to us because there's a ton of interest in trying to understand and predict pulmonary exacerbations in CF, and very few things have been found to actually be able to predict pulmonary infection. And so this was cool because it was sort of some early suggestive evidence that sinus health could potentially predict pulmonary exacerbation. Okay, so we got the patient clinical data. We measured cytokines and the concentration of different nutrients in the sinus wash. We also did 16S amplicon sequencing on 18 patients. And I'll just show you like a little snippet of what we saw with the microbiome, because that could be its own hour-long talk. But what we found, to make a long story short, in our adult CF-CRS cohort is that the microbiome of the upper respiratory tract looks a lot like what it looks like in adults in their sputum samples. And so here's an example. This is a taxa bar plot. The red is staph and the peach is Pseudomonas. And so each bar is a different patient visit and it's their microbiome at that time. And then the line that's over top is just the microbiome distance or dissimilarity from the first visit. And so what you can see is that in this case, this patient shifted from a largely staph dominant microbiome to having a bunch of Pseudomonas there later on. There's also the opposite example. So we can see sometimes folks will start with Pseudomonas and then end up with staph. Or there's examples certainly of people that just have tons of Pseudomonas throughout the study, and every kind of iteration in between, where in this last example where somebody's shifting back and forth between Pseudomonas and staph. So this microbiome data set kind of gives us a glimpse into the microbial community in which our Pseudomonas isolates are potentially evolving in response to, and it's largely staph and Pseudomonas dominated. Okay, and so then the last kind of set of samples here is we cultured Pseudomonas isolates from any time we could to get longitudinal isolates, and we did a whole bunch of in vitro phenotyping, so all those classic CF Pseudomonas phenotypes on a collection of over 100 isolates from these patients. And then we chose 6 patients and we did whole genome sequencing on all of the isolates that we could get from them, and we generated 67 really high quality Pseudomonas genomes here. Okay, and so now here's the questions that I'll address in the talk today. So for the past, we had one of our first questions was whether we would see these sinus populations as a mix of maybe environmental Pseudomonas that had been inhaled into the environment, or potentially because this was an adult cohort, maybe we would see something a little bit more similar to the lungs, where we would have maybe just one clonal population that had been evolved already. So the possibilities being, first, this, kind of a battleground in the sinuses of different Pseudomonas strains, fighting it out and evolving potentially in the sinuses, and then one population making it to the lungs, or maybe there's some amount of evolution has already happened in the sinuses. Okay, the second question is the present, so kind of our 2 minutes of the movie that we can see in our 2-year study. We wondered whether and how Pseudomonas might be evolving in the sinuses during CRS. And so historically, there is a lot of information about how Pseudomonas evolves during CF respiratory tract infections, but it's mostly done from lower respiratory tract samples from Pseudomonas that had been isolated from sputum. And so we really didn't know what to expect going into this with the sinuses. And then the future. So this is the last thing I'll tell you about if I have time today, is just a glimpse into some really new analysis that we're doing and wondering whether we can see some predictable genomic signatures of what are the first evolutionary steps that bacteria might take when they start to adapt and commit to living in a host environment. Okay, so starting with the past, are we going to see multiple strains of Pseudomonas in the sinuses or just one? Are they pathoadapted? To make a long story short, we used a molecular fingerprinting technique called RAPID, and we genotyped every isolate we had, and we found that most patients had just one single clonal lineage of Pseudomonas in their sinuses, so just like what we would expect in the lower respiratory tract for adults at this point. And so that's what this is showing you here. We did have some people that had two strains of Pseudomonas and one person that had three, but for the most part, just one clonally related lineage of Pseudomonas over time. And so then next we wondered, how pathoadapted are these clonal lineages? Is there any amount of evolution that happens in the sinuses? And so to do this, we undertook a bunch of sequencing, and one of the things that we did is when we picked the 6 different patient lineages that we would sequence all the isolates for, we took a hybrid assembly approach for the first isolate that we had from each of those 6 patients to generate a really high quality reference genome. So we did a combination of Illumina sequencing, which this is what the assembly would look like there. It looks like a ball of spaghetti. And we also did long read nanopore sequencing. And so we had the combination of the super accurate short read Illumina sequences plus the slightly less accurate but structurally accurate long read nanopore sequences, and together we made this hybrid assembly that was usually would finish into one contig or it would be circularized, so we could get a sense of, you know, what does the genome actually look like and what's the order of all the genes on the genome. Okay, and so when we did this, one of the first questions we had was we wanted to get a rough understanding of what we're working with. And so we wondered if we could make a phylogenetic tree and kind of put into context with a bunch of different lab strains of Pseudomonas that we're used to looking at. And so what I'm showing you here first is a phylogenetic tree that was built by this international Pseudomonas aeruginosa consortium, and they sequenced a ton of Pseudomonas isolates, and they found that there's 3 clades, and this first clade in blue here is the most common one, and our lab strain PA01 is present there, as well as a couple of other lab strains. This Liverpool epidemic strain is a CF strain. The second most common clade is this one here in orange, in PA14, which is another lab strain of Pseudomonas, is the kind of the representative of that clade. And then the third one is a super rare grouping here, and it's this PA7 strain. And so PA01 and PA14, we've done a lot of comparative genomics for this project with both of those isolates. They're both acute infection isolates. They're not thought to be host adapted. And so here, when we make the same tree for our own isolates, using just the first isolate from each of the 6 lineages that we sequenced, we can see that most of our isolates group with PA1, so they share a common ancestor with PA1. We did have one patient 33, that was more similar to PA14. We didn't have anybody that looked like PA7. Okay, so even though most of the isolates maybe shared a common ancestor with PA01, it would be deceitful to tell you that PA1 was a really great representative of our clinical isolates, because the clinical isolates have a huge accessory genome. And so that's what you're looking at here. This is a clustering dendrogram where every open reading frame is clustered with its homologs. It was made with a program called Anvio. And so you can see that there's a core genome down here that's highlighted in blue. So these are the genes that are present in all of the 6 strains, plus PA01 is also on here in yellow. And then this section here is genes that are not present in every single lineage. And then finally, this section here, this is the unique accessory genome. So these are the genes that are only present in one of our isolates. And to summarize just what we saw in these accessory genomes, so bacteriophage and IS elements were super abundant in the unique accessory genome. So these are the things that really drive differences potentially between strains. We also saw that efflux pumps and transcription factors are really abundant in the accessory genomes. And we also saw some diversity in different interbacterial competition genes like the type 6 secretion system and pyosins. It was very interesting to see for us. And so we did a whole bunch of phenotyping. So we looked for all the classic CF pathoadaptive phenotypes in our library of over 100 isolates. And what I'm summarizing for you here is the idea that we saw all of the classic CF pathoadaptive phenotypes that have been described for decades in the lungs and thought to have evolved in the lungs. We saw them in the sinuses as well. And so to summarize, these were phenotypes that suggested loss of acute virulence factor production. So, loss of swimming motility, loss of twitching motility, loss of protease. And also the transition towards chronic infection phenotypes. So we saw rugose small colony variants that overproduce biofilm. We saw increased antimicrobial resistance. We saw a reduction in cytotoxicity as we would expect as well. And so we saw all of these phenotypes in different combinations, though, in different patients. And so what you're looking at here is a phylogenetic tree of all of the whole genome sequenced isolates that we have from the study. And so you can, and then the reference PA01 is up here. And then this rainbow area right here is a phenotype key. And so if the block is filled in, that means that the phenotype is present in this isolate. And so you can see PA01 here, which is from an acute infection way back in the day, is expressing all of these acute virulence phenotypes as we expect. And then the different patient lineages of Pseudomonas have different degrees of pathoadaptation, it looks like, based on the phenotyping. So for example, patient 32's lineage really looks a lot like PA01, and it seems to retain a lot of acute virulence factors. And then in contrast, patient 33's lineage seemed to be the most pathoadapted. And so it was, these tiny mucoid colonies that were shiny and it just looked like what I've seen previously in the lungs of adults with CF. And then we could also start making predictions after we had all of that phenotyping data. We could predict how Pseudomonas might interact with airway epithelial cells at this point. And so in GenSlab, we have this polarized CF airway epithelial cell model. So we grow CF airway cells on this membrane support here. And then we can grow bacterial cells as a biofilm in association with the airway cells. And we can use this assay to measure cytotoxicity. And so what I did here was, this is TIR, so it's a measure of kind of the health of the epithelium in this assay. And we've got one isolate, the first isolate from each of the 6 patients that we did whole genome sequencing on, and an uninfected control in PA01. And so as expected, we know PA01 produces a lot of virulence factors and the TIR drops, so showing that the epithelium is unhappy at these time points. And then we see there's a range of different levels of drop of the resistance in this assay for the different lineages of Pseudomonas, as we expected, because it seemed that there could be different levels of pathoadaptation that had already occurred. And so just to kind of we see there's sort of this mirror effect of the increased cytotoxicity down here with PA01 in patient 32. And then in contrast, as we go up here, we have the most pathoadapted isolates like patient 33 here, as we expected, that hardly do anything in this airway cell model over the time course that we tested. And so from here, we could start making some guesses about maybe this patient 32 is a more recently acquired Pseudomonas isolate in the sinuses, maybe a more environmental like Pseudomonas, whereas the patient 33 isolate potentially maybe had been evolving in the host environment for a lot longer than patient 32. Okay, so that was the past. So we know in adults in our cohort, there's different Pseudomonas lineages that each patient has, and they all seem to have kind of different level of host adaptation, even from the beginning of our study. And then the next part of the study is the present. So we wanted to know whether and how Pseudomonas might be evolving in the upper respiratory tract of people with CF. And rather than take you through a laundry list of all the mutations we saw, I thought I would just kind of summarize how the isolates seem to be evolving. And so we have what we think we've identified is 3 different evolutionary routes to adaptive evolution in the sinuses. And so I'll summarize those now. So the first the first option that we saw was more maybe what you might think of as kind of a classic adaptive evolution through substitutions and small insertions or deletions. So lineages that evolved through this route had about 20 mutations per isolate per year. The second route was through hypermutation, and so these lineages had more like 200 mutations per isolate per year. And then the last route was mutations through mobile genetic elements, and so this was interesting. We had a lineage that had very few mutations, like substitutions or indels per year, and it was only until we did the hybrid assembly with the nanopore long reads that we began to appreciate the genomic rearrangements that were coming from mobile genetic elements. So I'll just summarize what we saw for each of these really quickly. So for the first route, this substitutions and indels route, here's an example of a phylogenetic tree of all of the isolates from one patient, patient 33, and you can see over time, these are the dates that we isolated the different isolates here, and you can see the tree shape kind of tracks with the date that the isolate was collected. And so another way to look at this data is to look at the accumulation of shared mutations over time. And so that's what you're looking at here. Each dot on here, so each isolate that we collected on each date will have one blue and one black dot. The black is the shared mutations or the fixed mutations that were present in that isolate, and then the blue is the singletons. And so what you can see is there's this linear increase in fixed mutations over time, as we might expect. And then we also can see a handful of singleton mutations that seem to get removed from the population over time. And one of the things that we thought we saw in this kind of classic route of evolution was the parallel evolution of iron acquisition strategies in the sinuses. And so what I'm showing you here is all the genes that it takes for Pseudomonas to make pyoveridin, which is a siderophore that it uses to capture iron from the environment. And it's thought to be this energetically costly to produce siderophore. It's a huge molecule and it takes a ton of genes to make it. And it's frequently lost in adult CF lung isolates. And so a number of times we observed the loss of pyoveridin production over time in our study. And so here's just one patient lineage where the earliest isolates all make pyoveridin, and then the last one stops making pyoveridin. We were able to hunt this down to a mutation in this PBDL gene that's required for the biosynthesis of pyoveridin. And we were also able to show statistically so this gene PBDL was mutated in 4 of our 6 patient lineages. We were able to show statistically that this was unlikely to be due to chance through simulations where we simulate mutations on the genome. And so what we think that we see here, at least for the pyoveridin and for a handful of other genes, is this parallel evolution across different patient lineages of evolution of iron acquisition and a couple other phenotypes. Okay, so the second route is hypermutation. Here's a phylogenetic tree where hypermutation emerged during the study, and so you can see this clade out here has this really long branch length from the earliest isolates of this patient, and we were able to hunt this down to mutations in 2 different DNA repair genes that occurred right here. And so what we found with hypermutation was interesting. It seems that hypermutation really accelerates the evolution towards loss of acute virulence traits in our study. And so I'll just summarize a couple of them here. So what you're looking at here is the loss of pyocyanin production. Pyocyanin is a redox active small molecule that's known to cause damage to airway cells, and it would be in line with our thinking about switching from acute to chronic infection phenotypes to lose pyocyanin here. Another phenotype that we saw, acute virulence phenotype that we saw being lost over time in our hypermutators was swimming motility. And so here you can see this isolate, the earliest isolate from patient 9 can swim, but over time, the latest isolate eventually completely loses swimming motility. That's another classic CF pathoadaptive trait that's been described over and over again as occurring in the lungs. It looks like we can see it in the sinuses too. And then, of course, the mutations that promote biofilm formation are a classic thought to be pathoadaptive trait. And we could see here's an example of a mutation in this isolate 56. This mutation fell in a gene that's a hybrid diguanylate cyclase and phosphodiesterase. It's involved in producing cyclic di-GMP, which would promote biofilm formation. And we could see these mutations in the region of the gene that might degrade cyclic di-GMP, leading to constitutively high cyclic di-GMP in this strain. So in line again with this idea of the isolates kind of pathoadapting over time towards chronic infection phenotypes. And then the third route was this was mind-blowing to see, I have to say, this proliferative insertion sequences that were just sort of riddling one of the lineages of Pseudomonas that we looked for. And so we saw a bunch of interesting mobile genetic elements, but the insertion sequences were the most interesting. And so insertion sequences are kind of the most simple form of a transposon. They just encode for a transposase and a couple repeat regions. And these repeat regions, I think, were why we weren't able to fully appreciate the severity of the insertion sequences riddling genomes until we took the hybrid assembly approach. And so when we did that, we saw this one patient lineage, patient 24, that had way more transposases than any of the other lineages. Here's PA01 down here has 6 potential IS elements in it. And so this guy had 92. And I just want to show you a couple of examples. I couldn't believe my eyes when I started looking at where are these IS elements falling in this genome. And so patient 24 has shared a common ancestor. This lineage shared a common ancestor with PA1. And if you look at the synteny, or there's great conservation in the kind of the order of genes on the genome. And so here's this locus where the REL genes are encoded. And so this is one of the quorum sensing systems that Pseudomonas has for coordinating group behaviors. And we could see this ISPA45, which is an insertion sequence, lands right in REL-R, the regulator, the transcriptional regulator of this quorum sensing system. And so we know that REL-R is involved in activating production of different acute virulence factors like rhamnolipids and phycyanin. And so this was really cool to see because you might expect REL-R could be mutated through substitutions, and that's been described before in CF, but it was really cool to see it get knocked out with an IS element. And just to summarize a couple others that we saw one of the other big quorum sensing systems in Pseudomonas, the last quorum sensing system. LAS-R also got taken out by an IS element. And then we even saw kind of overlap between our strategies 2 and 3, because one of our isolates had an IS element fell right in MUT-S, which is a DNA repair gene. You can see the super long branch length of that isolate because it became a hypermutator as a result of this IS element. Okay, and so to summarize that section of the talk, we think we identified at least 3 kind of evolutionary genetic routes that Pseudomonas can take in order to adaptively evolve in the sinuses and to evolve towards chronic infection phenotypes. And then the last part of my talk today, this is like super new, something we're just thinking about and just recently started digging into, and it's the kind of thing where you kind of need to just explore it as a possibility in order to see if there's something there. So we were asking the question of whether we can identify predictable genomic signatures of adaptation of Pseudomonas in the respiratory tract. And so when we were looking at our genomes, we started noticing some similarities between the genomic signatures of evolution that we saw in our sinus isolates and the early steps of genome degradation, as has been described in endosymbiotic bacteria that live inside of insect cells and intracellular pathogens. And so hear me out for a minute. So this is a figure from a review by John McCutcheon and Nancy Moran. They both study bacterial endosymbionts of insects. And so what they're showing you here is kind of this predictable course of over much, much longer evolutionary timescales than what we're studying in our 2-year study. This is like a multiple orders of magnitude longer evolutionary timescale. But what they're showing is that as bacteria evolve from being these free-living, non-host restricted bacteria that have this large and very streamlined genome, some of the first steps once they commit to living in the host environment is, there's genome degradation starts to occur. So they go from having very few pseudogenes, which are genes that have been inactivated in some way, to suddenly a lot of these genes become inactivated. There's a proliferation of mobile genetic elements, including insertion sequences. There's big deletions, and over time, these genomes get smaller and smaller. And so we became interested in this first step here and thought we saw some similarities. We don't see big differences in genome size in our lineages, but we do see some interesting stuff with pseudogenes and mobile genetic elements that we're starting to look into. And so what is a pseudogene? Pseudogenes form all the time as bacteria grow and divide and evolve, but they're typically quickly removed from the genomes of free-living bacteria. So here, what I'm showing you here is just kind of a cartoon of a gene on a genome in Pseudomonas. And there are many ways that pseudogenes can be formed. So you can have mutations that knock out a promoter, or that knock out the start codon, or a frame shift that leads to either a premature stop codon, or even a frame shift that leads to kind of a run-on ORF. And you get the gene in your evolved isolate is much longer than the homolog in your free-living isolate. So any of these things can lead to pseudogenes. It's not to say that every mutated gene is a pseudogene, but just that these are some of the hallmarks that you can start using to identify them in your genomes. And we can make some predictions based on how we think we have this, this range of pathoadaptation that's occurred in our different 6 different whole genome sequence lineages of Pseudomonas. And so earlier I had shown you that we think we have this patient 33 lineage that's super host adapted. And then in contrast, we have patient 32's lineage that really doesn't show any signs at all of pathoadaptation. We think could be maybe not have evolved in the host environment for very long. And we can make some predictions and think, okay, maybe in this patient 33 lineage, we might see some more signatures of early genome degradation. And in fact, that's what we saw. So what I'm showing you here is the length ratio of each gene in one of the clinical isolate genomes, either patient 33 or patient 32 relative to PA01. We did the same analysis with PA14 and we saw the same thing. We can see for the most part that most of the genes are the same length in our clinical isolate and in whatever reference we used. However, you can see patient 33 has a lot more shortened genes. It's pretty obvious on this side here. So it has almost 3 times as many of these kind of truncated genes that could be putative pseudogenes relative to the more environmental looking isolate. And so that was kind of a very interesting sort of a new line of inquiry in our lab. And I just wanted to summarize now, because I'm coming to the end of my talk, what are some of the other things that we're still kind of looking into and trying to see if there's something there, acknowledging that, our 2-year study or even our, lifetime of a CF patient is orders of magnitude shorter than the kind of the evolutionary timescale of obviously something becoming an endosymbiont. And when I'm not suggesting that Pseudomonas is becoming an endosymbiont here, but we do see some similarities. So we've seen these proliferative IS elements. I told you about that earlier in my talk. We see fragmentation in putative pseudogenes that seems to track with the level of host adaptation. We also saw reduced coding density that also tracked with levels of pathoadaptation, as you would expect. And then in work that I didn't have time to tell you about today, we've identified numerous nutritional auxotropies, which are another hallmark of free-living bacteria. Once it commits to living in the host environment, it becomes auxotrophic for a number of things like amino acids or nucleotide biosynthesis. And so we observed all these things, and we're continuing to dig into this as a kind of an interesting new perspective on the evolution that could be occurring in the upper respiratory tract, that maybe one of the steps of host adaptation in Pseudomonas is a significant amount of genome degradation that could occur in the sinuses, potentially even before the isolates reach the lungs. And so then just to summarize, I've shown you now that we think that Pseudomonas undergoes a significant amount of evolution in the upper respiratory tract before reaching the lungs. And so many of the mutations that we've studied and read about for decades using isolates from CF sputum, those selective pressures could potentially have, that drove those mutations could actually have been present in the sinuses, and the mutations could have occurred there first. This opens the door for that possibility. Of course, we need to continue with our study using paired sinus and lower respiratory tract samples in order to really figure this out. I told you about at least 3 different mutational strategies that we think Pseudomonas can use to adapt to the host environment, and especially I wanted to highlight that it wasn't until we did the combined Illumina short read and nanopore long read hybrid assemblies that we really began to appreciate the full contribution of IS elements and genomic rearrangements to this process. So a shout out to hybrid assemblies. And then lastly, we have some new data that's suggesting that Pseudomonas is undergoing potentially a significant amount of genome degradation as it persists in the sinuses. And this could be, seems like these genomes are really becoming dilapidated in some ways and could be an irreversible process. And so it leads us to asking more questions about what are the key early steps of Pseudomonas adaptation to the host environment, and how fast can this occur? And then finally, we have kind of my future interests as I continue down this route is I'm wondering whether we could identify specific mutations that are attributable to different changes in the airway environment. And so I have future plans of doing some experimental evolution in our airway cell model in the lab, trying to evolve our early isolates in the presence, say, of a respiratory virus infection, which we know changes the nutritional environment in a couple key ways. That's what Jen's lab studies, and seeing whether we can highlight the exact mutations that occurred in the presence of virus in our isolates. And so with that, I just want to thank Jen's lab and all of the folks over the years that have worked in this project. It's become huge. And especially Vaughn Cooper and Chris Marshall, who are incredibly generous with their time, and our clinical collaborators as well. And with that, I'll take any questions.
Thank you so much.
Awesome. Thank you.
Great
That was
talk.
great.
Thanks.
So I have one question from the first question from the chat, and it says, do you know anything about expression of pseudogenes and if frameshift mutations would be a mechanism to evolve novel virulence factors?
That's a great question. Yeah, so I know that there's interest in the pseudogene world. One of the things that if you want to really determine whether something's actually a pseudogene is you need to figure out if it's expressed. So are there mRNA transcripts that are made? How long do those last? Are they translated? So I don't currently know that. If we really wanted to go down the pseudogene route, that's something that I would need to do. I think certainly pseudogenes are known to occur in the process of evolving kind of new functions for genes. So one of the ways that something can become a pseudogene is if you have a gene on the genome and then it becomes duplicated, and then one of those copies of that gene can start acquiring mutations that maybe at first inactivate it and would put it in the pseudogene camp, but maybe through continued mutations in that gene, you might get a new function at some point. And so that's something that has been studied in other systems, not by me, in the pseudogene world, and it's super interesting. We definitely do have some examples of duplicated genes in our genomes.
So
Hopefully that answered the question.
Marvin, please go ahead.
Hey, Catherine, how are you?
Hey, Marvin, I'm good. Thanks for joining.
Yeah.
Nice to see you.
I have a question. When you talk about the IS elements, one of the bugs that's really known that IS elements play a big deal in the hospital is enterococcus. Have you looked at whether the degree of which you get this sort of IS element amplification in Pseudomonas, is it similar to what you see in enterococcus, or is it about the same that you see in enterococcus?
I don't know the answer to that. I will look into that though, because I don't know. I should look into that and see whether the enterococcus genomes
I think Mike's lab, when they sequenced like those 1,000 enterococcal strains, when they actually find ones from the hospital, they're seeing this massive amplification of IS elements, and it's thought to be one of the ways. The other thing that they're seeing, I think, and I was going to ask, is the IS elements also sometimes carry outward promoters, so outward running promoters, right? So that you're not inactivating genes. What you're doing is manipulating expression. Do you know do your IS elements have those? Or?
I'm not there yet. I don't know. I think that would be that would be amazing. That would be really interesting to see. If you could change transcriptional regulation of some downstream gene. It's uncanny where these things land. They actually look like most of them are landing within the coding regions of genes that we'd expect to be inactivated. So I kind of had stopped there with being fascinated with that and hadn't gotten to the point of looking transcriptionally now at the other genes that are around there and see if there's an effect.
Well, the other reason I was thinking is it gives you a little bit different way of thinking about it. Everyone looks at loss of function, but gain of function is obviously just as important. And I don't think anybody's gone after gain of function in Pseudomonas at all.
Yeah. That's interesting. Yeah, that's something I'll have to check out. I wonder, I mean, there are some tools I can think of that, can predict promoters. So maybe that's the way to go. Or there's not a lot for this ISPA 45. There's like one description of it, the paper where it was named, and that's it. But this one is, of all the IS elements that we saw in our study, that's the one that has this uncanny ability to land in like O antigen biosynthesis genes and
you could also look at transcriptomics and ask whether you're getting promoters that, because that you can map promoters.
Oh yeah.
That'd be the easiest way to do it. Then whether there's
we could probably just grow them in SCFM or something. And that's a great idea. With like promoters that lead outward.
Yeah, cool. Well, good talk.
Thanks. Yeah, thanks.
I have a question. So it's awesome that you found like these 3 specific mechanisms for this for the mutations. Which one is the most common one, and which one do you think was the most prevalent, or which one are all of these strains had all of them, or was it a combination? How is it?
That's a great question. Even though we sequenced a ton of genomes, or what felt like a ton of genomes in the study, this is really still a small study. So we only sequenced 6 lineages. So I don't know if I could really fully answer your question of which one's the most common based on 6 genomes, but we did see most of them did tend to have around 20 mutations per isolate per year, and they seem to be just like simple substitutions and things like that. So in that sense, the first strategy is the most common. We had 2 different lineages, we think, where we saw hypermutators emerge, and then all of the isolates did have IS elements, but the one that really blew my mind was just that patient 24. So one lineage where we could, I think, I see, just way more IS elements than the rest. That's a good question.
You said that you were basically the strategy is to test the sinuses and then try to follow up and see if you get the same in the CF, in the sputum, right?
Yep. Yeah, so that's ongoing. So we're taking a metagenomic approach for that. So the sinus, the study that I presented to you guys today was like our first foray into clinical studies in the lab, and also genomics. And so we did like single isolate sequencer genomes, kind of small scale. But the next version of this study is paired sinus and lung from every time point, and we have metagenomes, and will be interesting to see whether there's mutations that first arise in the sinuses, and then maybe the next time we sampled from the patients, we see them in the lungs. And that's what we'll really need to know in order to answer our question. Obviously, we need both samples, but we don't have that yet.
Yeah, because what I was thinking is that maybe they're already mutated by the time that so you have a strain in the sinus that's already pathoadapted. But then by the time that it makes it to the lungs, there's already other pathoadaptive strains, so it may not colonize,
be able to compete.
So,
yeah.
Right.
Yeah.
So it would be really cool to see if you see like this jump.
Yeah, that'll be interesting. I think there's going to be a lot of cool competition and interstrain dynamics that we might see in that data set, but we are only beginning to scratch the surface of it, so I'll have to stay tuned for that. Yeah.
Awesome.
Thanks.
Looking for other questions and see if anybody has any other questions. Meanwhile, while we're waiting, I have a question in terms of all of your study is showing is very nice that you see all of very common changes that we know that lots of people have done and found in patient samples from sputum at different stages of the disease. So there are lots of longitudinal samples for basically samples coming from lungs. And there are lots of ideas of, how this heterogeneity in Pseudomonas population happens, and different, maybe some were colonized in different parts of lungs. But you see already this heterogeneity is happening in the sinus. So what are your thoughts or comments on other than other bacteria, this heterogeneity in the environment, do you think this is all happening even before, the whole population is this mix and this jungle of different bacteria before getting to the lungs, or do you think it happens simultaneously?
Yeah, it's a good question. I see where you're going with it. So it's a tough one. So in this study that I talked about today, it was all adults, and they had all had functional endoscopic sinus surgery, which removes kind of a lot of the ultrastructure of the sinuses, and so it is kind of like one big gaping hole up there. However, we do still see, like you said, what seems to be different subpopulations and kind of regional heterogeneity. I think it could be present in both sites. So obviously, even if bacteria are first pathoadapting, taking those first steps in the sinuses, by the time they reach the lower respiratory tract, they persist there for a really long time too, and they do diversify. They diversify based on the regions in the lungs. We've learned that from a couple papers now, and so I think it could happen in both sites. It could absolutely happen in both sites. Yeah.
So basically, I think what you're suggesting, your data is suggesting, is like they kind of pre-acquire or pre-change and then go down the route of having more diversity happening in the lung, which is quite interesting because I think your future work would answer
Yeah.
it,
in a one of the things what we're really excited about the sinuses is not just to say like, oh, it happened there too, but the fact that the sinuses are so much easier to access than the lungs, and so there's this huge opportunity for coming up with interventions that could maybe, if these pathoadaptive phenotypes are really important for persistence in the lower respiratory tract, then maybe we can relieve, therapeutically relieve some of those selective pressures. Jen's lab has a huge interest in iron, and we know that respiratory viruses can lead to increased iron in respiratory secretions. And so, one of the possibilities is maybe if that ends up being an important selective pressure, then maybe we could use iron chelators or something like that in the sinuses. And so that's kind of where our excitement comes from, is the fact that we could
if you stop them going to the
dark
side,
or just slow it down,
becoming chronic,
slow it down with
very
some sort of if there's some sort of like iron chelation or something in combination with like the new triple therapies and things like that. I think we could really slow down this progression for for kids with CF, especially that are being born today.
Exactly. We have time for one question. I believe James Gurney. Yeah. Do you want to turn yourself? Okay, sorry. Do you want to unmute yourself, or?
I think you muted me, but I'm back up now.
Okay.
Hi, Catherine. Great talk.
Hi. Thanks.
I had a question about transmission. So obviously you've got quite a small sample size here, but part of pathoadaptation might be leading to increased likelihood of transmission between Pseudomonas. Because we see this when we have transplants lungs, right? People who have healthy, fully functional CFTR lungs will then get reinfected with Pseudomonas.
Yep.
My long-term worry is that we're with successful therapies, we're breeding Pseudomonas to become an ordinary lung pathogen for healthy people.
Interesting. So you're wondering about transmission in our study. So we didn't have any evidence of transmission based on the genotyping approach that we took with the rapids. Okay. So obviously not the gold standard approach for figuring out whether people were sharing strains, but it really doesn't seem like it, which is great for our clinic here. And of course, it was a small sample size. So, but I like to think hopefully there's no transmission in our clinic. They're very careful here. Does that answer your question?
Yeah, that does. Yeah. Thank you.
Okay, cool. Yeah, no, it's interesting. Yeah.
Cool.
Okay.
Well, I think we can. I would like to say that I think it's an awesome idea to try to figure out to compare how Pseudomonas is getting domesticated to be inside a host using this idea of symbionts. That's a pretty cool idea.
I've been fascinated by that literature, for a long time, and it's just uncanny. Once we saw the IS elements, it's like it's too much. It's like it still reminds me of it. But it is important to acknowledge that we're talking about 10 years here versus hundreds of thousands of years. No. So it's very different, but it's sort of the kind of thing where you have to look into it. If there's something there, you gotta do all the work to figure it out first, and maybe we'll find that it's nothing, but it's fascinating.
I'm all for finding alternative paths of science and then incorporating into your stuff, and you're if anything, it's just more fun, right? Look, and then something else will come up, and then you have another project. So
the more I study genomics, the more I appreciate the insanity of, say, the mealybug endosymbiont genomes and things like that. It's like the craziest. It's amazing that that they can figure out some of the stuff that they've figured out from those genomes because they're so dilapidated and
there's nothing. All right. Well, thanks again. It was great talk, and we hope to invite you to be physically in Georgia Tech at some point.
I would love to come down and visit sometime in the after days of coronavirus.
Well,
all right. Thanks
everyone for joining.
Thank you for everyone joining, and thanks, Catherine, for joining us today.
Thank you.
Bye.
Thank you.
Bye.
Bye. Gonna turn myself off. Okay. just going to stop the recording now.